have another kind of interesting piece of the course right now. I'm going to continue to talk about genetics, except now we're going to talk about the genetics of diploid organisms, which, apart from bacteria, most of the organisms, including us, we know are diploid. They have more than one copy of each chromosome, and so we'll go through a segment on this and also talk about mitosis and meiosis, these central processes of cell division and segregation of genetic material that underlie life as we know it. And then we're going to charge into a session on recombinant DNA and some of these technologies, PCR and various things that you see in the newspapers all the time. And then I'll finish up with a little section on the immune system, which is really, a few of you thought it was surprising that bacteria recombine. This, what I'll tell you in that system is almost like, it will feel like science fiction relative to what I've told you up till now, but it's an absolutely amazing system. So, but we're going to start today with genetics of diploid organisms. We're going to go back to how this was first understood. And most of you have probably heard of Gregor Mendel, who discovered this, and surely some fraction of you have run into an exposure to this topic before. But in keeping with what I'm trying to do in this course, could you guys just watch out there? Yep. I think I'm just going to unplug. Yeah, it's okay. I think I'm just going to unplug it for just a minute here anyway until we get a few more people in. You've probably heard of Mendel. Some of you have seen these different squares you might have memorized from a textbook or something like that. I'm going to try and see if we can go through this material up another level of sophistication, because again, as I'm saying, science didn't just come down from on high and end up with facts in a textbook. What's in the textbook is somebody's effort to sort of take a current state of our understanding, which is based on experimentation, and then come up with models. And what you're seeing in the textbooks are the models such as they were as of the time the textbook was submitted for publication. Sometimes they change even before the textbooks get out. But anyway, it's a process. And Mendel was one of the starting people who started this process, really a key figure and a guy with an amazing intellect. But before we start in, I just want to show you a couple of pictures, because these kind of knocked me over when I saw them. I don't know what kind of image you have of Mendel. You probably knew he was a monk, and he did something with peas, and he figured out this stuff about genetics. And most people carry around an image probably somewhat like this sort of romanticized drawing. He was a monk, all right, but it was at a monastery, an Augustine monastery in Brunno in Austria, that was a very major intellectual center. They even published a scientific journal. They sent Mendel off to Vienna to go to university. While he was there, he studied physics, math, as well as botany. So he had, in many ways, a background that was very similar to you guys. Very heavy on sort of the quantitative, physical science, mathematical sort of background. And then he went on to do some experiments in biology. And I think you may even get a sense of this if you see a picture of what Mendel actually looked like. Here's one picture of him. But the one that really blew me away, I have a picture of the monks. Now just think of whatever image you had of the monks that Mendel was at. Well, there's a picture of them. To me, they look like nothing so much like a group of university presidents or something sitting around for a portrait. And he traveled very widely. He was on his way to London here. Here's a picture of him with a group of people on his way to, I think he was in Paris on his way to London. So this wasn't a little isolated monk in a garden who stumbled across stuff. It was a rather sophisticated guy going after some interesting problems. And this was the garden in which he did his experiments. There's a picture of it. So this was pretty straightforward experimental setup for these really amazing things he did. Now, so with that kind of background, Mendel was interested in the problem of inheritance. And people have been aware that traits were inherited. That was the whole principle of domesticating animals and domesticating crops, that if you took parents with certain characteristics and crossed them together, that the offspring then had the traits that were associated with the parents, and so people had been able to sort of get better domesticated animals or better domesticated crops. But up till then, it was sort of this mixing was thought of sort of like blending liquids. Stir together some green and some red and a little this and that, and it all stirred together. And as you'll see, what Mendel's one of his great insights was that sort of it wasn't like mixing liquids. And to study this problem then, he picked a system, but he didn't just, it wasn't that he was just fiddling around with peas. He was a pretty sophisticated guy, and he picked peas as an experimental organism for three reasons. So here, Mendel, and so why peas? Well, one, they were easy to grow, and that's still a major consideration of any model system that you want to use in science today. If it's really tricky to grow, it's very hard to do work with. It was easy to pollinate in a controlled way. Just the structure of the pea flower makes it very easy to make sure to either put the pollen right on the pistil of that same flower, which is a kind of self-fertilization, or to make sure that the pollen goes from one flower to another, in which case you're doing cross-pollination. And the third thing, and this was the really important thing, was this system had been worked on before, and there were a number what were called pure breeding lines. If he just grabbed peas out of the wild, it'd be sort of like me starting to do genetics by crossing a couple of you guys. You know, we'd get offspring all right, and we'd cross those offspring, and we'd keep getting things that people that look different and different, maybe something like the parents. But what had happened with peas is people had taken a pea and then they continually inbred it until it finally sort of settled down. And it like always had white flowers. It always had wrinkled seeds. It always had whatever the particular trait was. And so here was here, I think I showed you this slide earlier. This shows two things. You can see there are smooth seeds and there are wrinkled seeds. And it may be a little hard to tell, but in this light, but there's sort of two colors here. One's kind of greenish and one's sort of yellowish. So there you see two of the traits right there. There were also flower colors and height and other things that had these different had the characteristic. They were pure breeding, and you could, every time you took that line and took if you self-crossed it and put out its progeny, you'd see the same characteristic each time. So this was the system that Mendel started to study this problem of inheritance, and how did this blending come together when two organisms brought pollen, and or if it was a human, it would be other animals like us, it would be sperm and egg, but somehow you did something that ended up giving you an egg that got fertilized, and out of that came the progeny. So what Mendel did, the term that's used, they say he carried out a cross, because this is genetic speak here now. So he took pollen from one plant, fertilized another, used it to fertilize another plant, collected the seeds that came out of this, and examined the progeny. And I think an interesting way of thinking about this is as a Europe project. You'd come into Mendel's lab and wanted to do a Europe project. It was pretty easy. I could probably show you all the techniques you needed to know. This is it. We show how you pollinate, we collect seeds, and then we look at the characteristics. So it's just some fairly simple manipulations and some observational stuff. So let's suppose you know, you're doing Mendel's thing as a Europe project, and let's just see where that would take us. So what Mendel did to begin with, he took one of these pure breeding lines that was smooth, or all abbreviated as a capital S, and he pollinated it with something that was wrinkled, or I'll do that as little s, and then he collected the seeds from what's known as the first generation, starting with something like this, and geneticists use the term F1 for this first generation in a cross like this. And what he found was all the seeds were smooth. So the wrinkled trait had, if you will, disappeared. That's your first Europe experiment. You do. Time to submit to Nature or Science or something, write a little paper like Watson and Crick that turns the world on its end. What'd you do next? No gels, no whitehead sequencing facility. Anybody got any ideas? I've showed you all the techniques that he knew about. Pardon? Cross it again. What you thinking? You think the trace disappeared, or do you think it's hiding? Might be hiding, right? I don't know what he thought, but I think that's a reasonable way to think about it. He's probably trying to figure out, did this wrinkled trait just disappear from the face of the earth, or is it hiding in those first generation seeds? So let's put that up here. So that's exactly what he did. So he took these seeds now that were the smooth F1 . These are not the same smooth as the parental up here. Just to make clear, you guys understand, these are pure breeding. These are the ones that people have been breeding for a long time. This one and this one. In this case, even though I'm drawing it as circles, this is the smooth F1, and this time when he did the experiment, he looked at the second generation, or the F2 generation, as a geneticist would call it. And what he found, he got some smooth, and he got some wrinkled. So the wrinkled trait reemerged in the F2. So it wasn't really gone, it was hiding. Okay, time to submit to Nature Science. So, got it, we figured. He didn't try and publish it at that point. He did something else. They said he was kind of like, same kind of background you guys have. Anybody think what he might have done? Could cross it again. Something else he did with this experiment, though. Pardon? Well, you're thinking of new experiments. He's got a little data processing he could do here. What's that? I'm not able to hear, sorry. Statistics, okay. That's good. Maybe, like, I'll simplify it even slightly before that. I've got some of each. Count them, right, exactly. So that's what he did. And I think the numbers were, if I remember, 5 for 74 and 1850. So, what should I do now? Ratio, absolutely, right. We could count another million of them, but that probably would be not terribly productive. And what he found out was when he did that, he found that he got a ratio that was pretty close to 3 to 1. And that was sort of what he found out from this by doing this sort of thing over again was a pattern. A trait would disappear in the F1, trait reemerge in the F2, and the two traits had this ratio. The ratio of the two traits would be about 3 to 1. If you don't think this is sort of like a Europe project or something, that's a page out of some of Mendel's actual notes while he was doing his crosses. And what he did next then was to take some other traits. Yeah, sorry. Whoop, excuse me. F1, thank you. Remerged in F2. So he took some other traits, white and purple flowers, tall, short, found that there were at least certain other traits. It didn't work for everything he studied, but with some of them, he could see the same pattern. One of the traits disappeared. It reemerged in the F2, and when he counted them, he'd find that the trait that had reemerged was at 1, and the other there were 3 times as many. So he'd seen a pattern, and all he's done at this point is to cross flowers and count the progeny. So at that point, Mendel tried to explain his data. So he had to now take the next part of the scientific process. And what's kind of nice about thinking about Mendel in this sense is we're not overwhelmed by complicated techniques. You can see, I think, the scientific process in its very bare bones thing marching along. So now he's got some data. He's quantitated his stuff. He's found it reproducible. It's not only for seeds. It seems to be some general feature. And the thing about this, I guess, I don't know what he thought, but it seems to be likely that he could see that this didn't fit very well with the blending idea, like you pour together 2 liquids and you stir them all up. Instead, he really made this monumental leap in thinking that genetic information must come in some sort of particulate form, come in particles or units or quanta, if you want to think of it, if you're a physicist. We know those units as genes right now, but that may seem, we grow up with it right now, but to go from the idea that genetic information was kind of like 2 liquids blending to the idea it was little particles, it's just about the same kind of leap as thinking that energy comes in particles instead of like a continuous sort of thing. So that was the kind of insight that Mendel had, and then so genetic info comes in particles, units. As I say, we now call these genes. And then if that was what it was, then he started to think about these traits as particles that had a different character associated with each of them. That would mean there'd be one particle that was big S, and that was smooth. And there was some other particle that was specified the wrinkled character, and we'll call that small s. So what was happening in these crosses then, he was now mixing particles instead of liquids. Now again, I don't know how he got to how many particles there had to be of each per organism. It could have been anywhere, from 2 upwards. He had to have 2 in order to explain the sort of stuff he was working with. No reason he couldn't have thought of 12 or something, but I assume start with the very simplest thing you can number that you can think of and see if you can make this work. So what he hypothesized then was that each organism had 2 copies of each of these particles. So 2 copies of each particle. So this would mean that there were 2 types of particles, so there would be the S and the smooth. So he could get 3 types of things. He could get one that was both big S or smooth, or you could have the ones that were both the 2 little s's, and these would be wrinkled. Or if you had the other combination, what he figured out to fit with this model was these would have to be smooth. That would have to mean that one of them is dominant over the other when you put them in combination. So in this thing, the big S would be said to be dominant, and a little s would be said to be recessive. And there's another little term here that I'm going to introduce because it'll help us talk about this stuff over the next few days, something terms geneticists use all the time. Because these are both have 2 of the same, they're said to be homozygous, 2 of the same, and this one with 1 of each is said to be heterozygous. Okay. So there's, I think, the sort of setup for Mendel's model. He had to contend with one other issue, though, and that was if every organism has 2, and 2 parents get together and bring some, and each donate something, unless you did something, the offspring would have 4. When those offspring got together, the next one would have, twice as many, and so on. So from probably, I don't know whether it was just from first principles, but I would imagine he figured out that if organisms had sex, as with pollen and whatever, or egg and sperm, that something had to be done to get around this problem of an ever-increasing number of particles. So what he envisioned was that when there were specialized cells for sex, and that they halved the number so that the sex cells would have half the number of particles, so that when each parent donated 1, you'd be back up to 2. Pretty simple, straightforward thinking once you've gotten the idea that these things are coming in particulate form. Okay, so with this, could he now explain his results? Let's do it over here. So what happened in the first cross? He had this smooth pure breeding line crossed with, and so the sex cells from this, each one would have been a big S, and the sex cells from each one of this would have been a little s. And as you recall, what he got was all smooth, right? Remember? So if we try and figure out what's happening here, a way of representing this would be to think what happened if all the combinations that you could get. So if we paired them in all possible ways, then every combination would be identical in that first cross. 1 from one parent, 1 from the other. And if one is dominant over the other, it's going to look like this. This is really the word I introduced you to. That's the genotype. That's what's going on down at the genetic level. And what you're seeing up here is the observable characteristics of the organism. That would be the phenotype. So then, what would happen then with this if you crossed the the F1s? Well, as you recall, they were smooth, but he was now seeing them as being like this. So that means that the sex cells that are generated from this, each one will generate one big S and one little s. And then if you put them together to see how this would work out, well, this one's two big S's. This is a big S and a little s, a big S and a little s, and two little s's. So what he's got here is over here is SS in a ratio of 1 to 2 to 1. But when you look at the phenotype, what would we expect? Well, this would be smooth. Big S and little s, that's smooth. Big S, little s, smooth again. And two little s's, that's wrinkled. There's his ratio of 3 to 1. Has he proved anything? It works? Beautiful. Model must be right? What do you think? You ready to publish? Should prosecute. Why is that? Why did the model work? Has the model predicted anything yet? No, it works because it describes the data. Sort of like fitting, to some extent, it's like fitting a curve. You fit it, but you don't really know yet. Of course it's going to work, because if you got different data, you would have had a different model. So you're putting your finger on a really important point, and that is you can do an experiment, you can get data. It doesn't have to involve DNA sequencing or a fancy technique. You're getting data out of a biological system. You've come up with a hypothesis that explains it, but of course it's going to explain it, because you wouldn't publish a model that didn't explain your own data. But what he hasn't done is tested it. Will his model predict the outcome of something that he hasn't already done? So the suggestion was that he should carry out another cross. And that's what Mendel then did. Again, he didn't, this is what he had to work with. He could cross and he could count. And he could do some calculating and some thinking, but those were the techniques. That's why I really like thinking about this, because you can sort of put yourself in his shoes. So what would you guys like to cross? We haven't got much, right? One he did, one cross he did. He crossed the F1 with the homozygous dominant parent, the pure breeding line. So he's got this smooth that's a big S and a little s, and he's crossing it with something that's two big S's. So the sex cells that you'll get out of this are this. So if you set this one up and see what happens, there's the two, you'll get two big S's, big S, little s, two big S's, little s. This is sort of uninformative. If you want to look at your notes afterwards and have this be consistent, let me just flip this slightly. Let me put the two big S's up here, and this is our F1 down. That way I'll be following the same pattern as I did before. Okay, so there we are. In any case, they're all smooth, but that's not particularly helpful. He's seen that result before. It hasn't really proven out, given a sort of unexpected result that's predicted by his model. But he tried another one that's very, very similar, and this is, in this case, he crossed the F1 with the homozygous recessive parent. This is a really important cross in genetics, and the reason because it's so important, it's given a special term. It's called a test cross. Let's see what happens with this one, because this one's more interesting. So we take the F1, so this is the F1. He's now crossing it with this homozygous recessive, so a parent that's got two particles that are little s. And so what will the sex cells look like this? A big S, little s is before, two little s's here. So if we set up this as we've done, there are the sex cells from this F1. Here are the sex cells from the homozygous recessive parent. Up here we get a big S and a little s for each of those, but here we get two little s. So if we look at the phenotype, if you're out in the field or out in the garden or sitting in the kitchen after you brought the seeds in or wherever he was working, what would you see predict you'd see in a cross like this? Well, these would be both smooth, but these would both be wrinkled. So here at the genotypic level, we've got a ratio now of 1 to 1. And here as well, there's now a ratio of 1 to 1. So there you have a result that you haven't seen before. And if you do that cross and then get that result, again, it doesn't prove your model. I mean, scientific proof it's never a QED. Somebody can always come up with an experiment tomorrow that disproves it. It tends to work more. You just keep shoveling on evidence, and finally someone says, enough, I believe you. So this was at least a test of the model, and the model survived this test. Now, he did one other experiment. We think of a combination of the things that we've got on our plate right now. Is there anything else we might do you can think of? He did another cross. Pardon? Two of the heterogeneous ones. We did the F1s against each other. That's where we got the 3 to 1. We've already crossed the F1s, but I like your idea. What if we took the F2s? In this case, it's going to be pretty complicated because they've got this 2 to 1 to 1. But one of the things that you can do with peas is you can self-fertilize them as well as cross them with their neighbors, because they've got both. They've got the ability to make the eggs that'll become the seeds, and they have the pollen, which would be equivalent to the sperm. So they've got both. So as long as there's some plants that you can self-fertilize and some you can't, one of the really nice things about peas is they have the property that you can self-fertilize them. So another kind of experiment that Mendel did then was to self-fertilize. It's another test of the model. Self-fertilize the F2s. Well, the model predicts, if we look back over there, that what you'll have there is a mix of things in a ratio of 1 to 2 to 1. So if you were to take seeds from that F2 and then cross them with themselves, if you could figure out all the different outcomes and then sum them up, you'd have a prediction for what this model would suggest, right? So let me just take you through the pieces. Let's think about a quarter of them, according to the model, a quarter are that. So if we cross those, what we should get is all wrinkled, because the only thing we're dealing with is the wrinkled trait. So that would be a quarter of the F2 seeds would be expected to give that outcome. So 3/4 of them are smooth, but it's tricky because there's 2 types of them in there, right? So of these, 1/3 are this, and 2/3 have that, right? So what would happen if we thought about each of those individually and thought about the outcome? Well, if we take the SS type and we self-cross them, what we're going to get is all smooth, because all we've got are the trait in the cross are the traits for the smooth characteristic. And if we take these guys and we've self-crossed them, well, we've already done that. We know what we will get is we will get smooth to wrinkled in a ratio of 3:1. So again, now you could now sit down with that and figure out in total what you would predict in terms of smooth and wrinkled if you self-crossed the F2 and if the model was correct. So that was basically Mendel's first Europe project, or the end of the first term or the first year or something like that. And he did publish those results. They were published in 1866, which was the same year as those, about the same time, I think, as those results that Pasteur was publishing that I told you about earlier on. So it was a very impressive and you know, in retrospect, a truly major intellectual leap. But it had almost no impact at all on the world. And there's a theme here that I've sort of tried to hit up, you know, several times, and we saw it to some extent with the early work on DNA, that someone can get evidence for an idea, but if the scientific community isn't ready to accept it, it can take quite a while before that idea becomes credible, even if it's correct. I mean, the data was there that DNA was a genetic material quite a bit before the general scientists thought it was, and it just seemed like it was too simple a molecule, too boring a molecule to be possibly able to encode anything. And the same sort of thing happened here. It was some geneticists picked up on this, but not until about 1900, and they all cited Mendel's results. But the reason was some stuff had happened in between. What Mendel had essentially up to here was somebody mentioned statistics. It's basically a statistical argument that this is what's going on. The problem was there weren't any particles. These were just no evidence, physical evidence for the particles. They were just imaginary units, sort of entities that he dreamt up in his head. And as I'll show you in the next lecture, shortly after this, people looking through microscopes began to see chromosomes. And I'll show you some movies at the start of next thing where you watch a cell divide, and you can see these things that when the cell divides, they split in two and equal numbers go to both sides. So that you could sort of get the idea that there were physical entities that had these sort of characteristics that Mendel was suggesting of segregating to offspring. So another problem that Mendel had was sort of, you can sort of see one pattern here, and that is he found this thing with, say, the wrinkled and smooth seeds, but to make sure it wasn't just a peculiarity of seed shape, he went and looked for other traits, and he found those. So he generalized his result. He saw a pattern. Once he saw the pattern, then he felt he was on firm enough ground to frame a hypothesis. So in the same kind of way, he wanted to extend his results from peas. Now, sometimes there's bad luck, too. So there was a botanist who studied weeds who was very much against Mendel's stuff, and he suggested that Mendel study this in weeds. And the problem was these weeds could form seeds without ever fertilizing. They could just, basically self-clone and put little copies of their own genetic material into a seed. It's actually interesting. I learned citrus trees can do this too. Citrus genetics is very complicated because they not only produce progeny like we're talking, but they also, some of them are just vegetative clones or copies of the parent. Apples don't do this. Johnny Appleseed would have had a different sort of outcome. So that actually didn't work. He couldn't reproduce his results in weeds. So his next idea was to try bees. And in fact, I found a picture. Oops-a-daisy. Bring it up here. Oh, or did we lose it all? Oh, okay. There were a bunch of beehives that Mendel designed to try and extend his result. But he had bad luck again. Because we won't have time to go in this, but some of you know, if you've probably heard of bees have a hive, there's a queen, there's a lot of workers, and the male bees are drones. And we hear that. They don't contribute to the work of the hive. They're just there to fertilize the queen, and that's their role in life. And so he could get the male bees and get the queen, but the problem was male bees are not diploid. They don't have two copies of everything. They're haploid. So when he tried to do crosses with bees, he didn't get the expected result either based on his other studies. So Mendel actually didn't, his work sort of stopped with the peas. He tried it and it sort of petered out, and it took, as I'll say, quite a few years for it to do it. One other interesting little piece of history was that Charles Darwin, who was turning the world upside down with his ideas about evolution, would have really liked Mendel's results because the kind of evolutionary thoughts and selective forces that Darwin was envisioned wouldn't have worked well with this sort of stirring liquids model, and it worked very well with the particulate model. And Mendel sent him a copy of the manuscript, but later on, when they were looking in Darwin's effects, they found the envelope from Mendel but unopened, so Darwin had never seen it. So anyway, as I said, I'm trying to get more across to you in this thing than just facts out of a textbook. And it's an experimental science. It's done by real people. Models are made. Models are tested. If there'd been a textbook, it would have taken quite a long time for Mendel's results to get in here. We still have them in there because they've survived till now. But there's all kinds of other factors, the preparedness of the community to accept an idea, and sometimes just bad experimental luck. He was very lucky, picked the right system at the beginning, but didn't do so well on the next two in terms of good experimental models. Okay? So we'll talk all about meiosis and mitosis next time.